All right, guys, we're going to start part 2 of our chapter 10 activities. This section it's not too big. We're just going to talk about how DNA makes copies of itself. We know that we need to make copies of DNA before we give it to a new cell. If we didn't do that, our new cells wouldn't have any genetic material, any information to use. Before we go into that, though, just a little review from last week. We talked about our DNA model, our double helix of DNA, which we see pictured here. It looks like a twisted ladder or a spiral staircase. We talked about some things that make up that DNA ladder. We said the sides of that ladder are made up of sugar and phosphate. They help hold the sides of that ladder together. And then in the middle of that ladder, this is something we focused on a bit last week. We have those complementary base pairs, those nitrogenous bases in the middle that have different letters. In DNA, those 4 different letters are A, T, C, and G. In our picture on the bottom of this slide, we see that that DNA ladder has been untwisted to show us how those nitrogen bases kind of pair up. We talked about this last week, and it's really important to remember, so we're reviewing it here again in this slide. Whenever we have these bases matching up, they always match up in a certain way. The letter A, that base always matches with the letter T. So A and T, we say they go together like apples and trees. The same thing is true with letters C and G. C and G for cars and garages, we say they go together in the middle of that ladder. Those pairing rules are important because they help give organization to our genetic code, and that's something that every DNA molecule is going to follow. So those are the basics that we went over last week. We'll also this week talk about how we make copies of that DNA molecule. So when we make a copy of DNA, it's called DNA replication, making a copy of something. Just like when we were in class and I would make copies of things for you guys like worksheets or notes. I replicated something. I made a copy of it. The same thing happens for our DNA so that every cell gets the same copy of information to use. So this is the process by which DNA is copied in a cell before a cell divides by mitosis, meiosis, or binary fission. So it's important to note that this happens before the cell divides. We can see in the picture over to the right the way this is going to look. When we replicate DNA or make a copy of it, what we're going to do is we're going to untwist that DNA ladder. It almost looks like you're like unzipping a zipper on like a jacket. Okay, we're untwisting it so we can separate those base pairs in the middle. And we'll do that by using an enzyme called helicase. And we'll go through those steps on the next couple slides. So there are 3 main steps in replication, and we're going to walk through this in pictures on the next slide as well. But our first step is that this enzyme called DNA helicase will come into the DNA molecule and kind of separate it. It will separate the hydrogen bonds in the DNA strand that holds it together, and it untwists that ladder. It creates something called a replication fork. So for step 1, a very important enzyme called helicase breaks up that DNA molecule. For step 2, we have another DNA enzyme called DNA polymerase. This is going to attach the DNA molecule, and it's going to add complementary nucleotides or building blocks to form new DNA strands. So we're going to have a second enzyme come in and start to build a new DNA strand. For our third and final step is called release, and we're going to have that DNA polymerase enzyme release itself. It's going to leave that DNA molecule once it has made copies And now we're going to have two identical DNA strands as long as no errors occurred. Something else to add to this picture as well, those two new DNA strands you might notice in this picture are shown in two different colors. So we have half of the DNA strand that's in blue, half that's in green. That shows us that for this DNA strand, half of it is actually from the original DNA strand. That's the blue part of each of those. And then half of it is actually brand new that's been added on by the DNA polymerase. So we call this semi-conservative replication. That means that half of the DNA strand has been kept from the old one, and then half of the new DNA strand has been added on by new pieces by that DNA polymerase. So that's what we end up with. We end up with half of the DNA strand that's old and half that is new. It's like if you split a ladder in half and then you added a new side onto each of those halves of a ladder. You would have half of the ladder that's new and half that is old. Replication is going to happen a little bit differently between prokaryotic and eukaryotic cells. Prokaryotes, if you remember, they don't really have a whole bunch of chromosomes. They have like one circular chromosome, one DNA loop. So replication is going to have a single origin or a single starting point, and it's going to go around that DNA loop in a circle and make a copy of it. There'll be two replication forks on that, two places where the DNA is being unwound. For eukaryotic cells, we have many long but not circular chromosomes. Remember, in each of our body cells, we're going to have 46 chromosomes. So we'll have to make copies of all of those. Replication is going to begin at many starting points or many origins and have many replication forks. So it's going to be a little bit more complicated in eukaryotes, just like everything else. Eukaryotes are a little bit more complicated than bacteria cells. So replication is going to take place maybe longer and have more starting points where it actually begins. So now we're going to talk about how DNA errors occur. Usually our cells are very, very accurate in copying DNA. Okay, they have to be. But they are copying such a large amount of information that errors are going to happen from time to time. This would be like one of us trying to copy a 1,000-page book and write it down word for word. We're going to make some mistakes because we're not perfect. Our cells are going to make mistakes because they are not perfect. There are billions and billions of letters in our DNA that have to be copied, and our cells occasionally are going to make an error in copying them. Errors occur about 1 in 1 billion paired nucleotides. So for every billion letters in our DNA, we're going to find usually an average of 1 mistake. When you have so many letters in there, that can add up. In order to avoid this, we're going to have some proofreading that our cells try to do. We have some enzymes that help check for any spelling errors that could be in our DNA. For example, if we found that a C was bonded with a T instead of a C being bonded with a G, that would be an error that would happen in our DNA. And we have some enzymes to kind of proofread that and make sure that it's okay, but even those enzymes sometimes still make mistakes. If that mistake happens, that means that we have new DNA that's going to be different than the original. So let's take a look at our picture over to the right. Up on top, we have our original DNA strand, and they only gave you 3 letters there just to keep it simple. Okay? Now, we show an example where we have 2 copies that have been made from this. On the bottom left, we have a correct copy, which is the exact same as the one up on top. We have a C and a G together, a G and a C, and then T bonded with an A. Now, on the right side, on the bottom, we have a mutation that has occurred. Instead of the first letter pair being CG, it is now TG. And we can see that, that is going to change that DNA molecule. That gives us a different letter in that code. So that could change a gene, and that change could be good, or it could be bad, or it could really not make a difference at all. We'll talk about some of those types of mutations, but the big idea is that the mutation always changes the DNA. That new DNA strand over to the right is now permanently changed. It's no longer going to have a C and a G. It's going to be a T and a G. And when that new DNA makes copies of itself, we're going to see that mutation be carried on. And more mutations could occur and add on to that. So that's how we get different mutations and different genes showing up in organisms that were not there before. So a mutation is any change in that nucleotide sequence or that letter sequence of a DNA molecule. And sometimes this is caused by random errors, like a random copying error, sort of like we saw on the previous slide. However, there are some things that cause mutations to happen or make it more likely for them to happen. Certain types of chemicals, like cancer-causing chemicals that we might find, for example, in cigarettes and alcohol. Some of those chemicals can make it more likely for mutations to occur. Radiation is another one. Not just like radiation from like a nuclear bomb, that's really bad, but also ultraviolet radiation or UV radiation from the sun. We talked a lot about skin cancer this year as being one of the types of cancers that's still on the rise. A lot of people don't think about protecting their skin as much as they should, even myself included. I'm outside a lot, and I don't always think about putting sunscreen on. That radiation that's coming down from the sun has been shown to increase the likelihood of different mutations happening inside of my skin cells. And those mutations can sometimes lead to like skin cancer, like we see pictured on this person's nose towards the left. Now, it's important to note that mutations don't always have to be bad. Sometimes mutations can be favorable or they can be good. For example, a mutation might give a rabbit a different fur color that helps it blend in more. That would be a good mutation because it's going to be able to hide from predators more than rabbits that have a different fur color. So that might be a mutation that would help that population survive, and we would see more and more of that mutation showing up. However, mutations can be harmful, such as cancer, for example, and then sometimes a mutation doesn't really make a difference. It's not good, it's not bad, it's sort of just there. So those are kind of three categories of mutations that we have. So that wraps up part 2 of our DNA chapter. So you'll be getting some questions and some small assignments about this, dealing with DNA replication, the enzymes that are involved, and then dealing with those DNA base pairs and what can happen when mutations occur. So please make sure you review this video. Make sure that you review your PowerPoint that I've given you for Chapter 20. You'll be able to use those on your assignments as we move forward.